Welcome to nonstopneuron.com, where learning medical concepts is as easy as watching cartoons. In this video, we will see the transport of glucose, amino acids, and urea in renal tubule. So let's start with glucose. First, let's take a look at its filtration at the glomerulus. Its concentration in plasma is about 100 mg per deciliter. Or in simple words, 1 mg per milliliter. And it's freely filtered at the glomerulus. So at the glomerular filtration rate of 125 milliliters per minute, its filtered load is 125 milligrams per minute. In short, normally, 125 milligrams of glucose enters the tubule every minute. Now coming to the reabsorption part. The proximal tubule plays the main role in this. At normal blood levels, it is reabsorbed almost completely. So very trace amount is excreted in urine. Now let's see the mechanism of reabsorption at the molecular level. The reabsorption occurs by the transcellular route. Entry across the luminal membrane occurs by sodium-glucose cotransporter. It is a form of secondary active transport. Passive entry of sodium into the cell energizes the transporter. This in turn drives the entry of glucose. Exit occurs by passive diffusion by glucose transporter 1 and 2. So, almost all the glucose is reabsorbed in this way. Now, there are a few more concepts related to glucose reabsorption. In them, we will be dealing with a filtered load of glucose a lot. And earlier we saw that the filtered load is a product of its plasma concentration and GFR. That means, the filtered load of glucose can vary with a change in plasma glucose concentration, or GFR, or both, right? However, changes in glucose concentration are more common than changes in GFR. So to keep the subsequent discussion simple, we will assume that GFR is fixed at 125 milliliters per minute. So filtered load of glucose is directly proportional to the plasma concentration. In short, in our subsequent discussion, we will consider changes in plasma concentration only, not in GFR. On that note, let's see the concept of transport maximum. We have seen that normally 125 milligrams of glucose is filtered every minute, and almost all of it is reabsorbed. But you know what? Tubule can reabsorb even higher loads also. However, this increase in reabsorption can occur only up to a limit. Beyond this limit, there is no further increase in reabsorption. This limit is called the transport maximum. Now let's see the mechanism behind this. Let's say that this nephron has this many transporters. From them, to reabsorb 125 milligrams glucose every minute, these many transporters are sufficient. The remaining are inactive at this much load. If the blood glucose level rises, more glucose gets filtered at the glomerulus. At higher glucose levels in the tubule, more transporters come into action to reabsorb additional load. In this way, as more and more glucose gets filtered, more and more transporters keep coming into action. So the reabsorption keeps on increasing. So glucose does not appear in urine, even at higher blood glucose levels. But this can occur only up to a limit. Once the transport system gets saturated, there can be no more increase in reabsorption. So the additional load starts getting excreted in urine. Now let's plot a graph of plasma glucose concentration versus filtered glucose and reabsorbed glucose. First, we will talk about the filtered quantity. We have already assumed that GFR is fixed and filtered glucose is directly proportional to plasma glucose, remember? So, as plasma glucose rises, filtered glucose rises proportionally. So we get a straight line. Now let's draw the line for reabsorbed glucose. Initially, when plasma glucose is on the lower side, all the filtered glucose is reabsorbed. That means reabsorbed glucose is equal to filtered glucose. So in the initial part, the line for reabsorbed glucose falls right on top of the line for filtered one. The same applies till we have spare transporters. but once the transporters are saturated, reabsorption reaches to its maximum. So there won't be any increase in reabsorption with a further increase in filtration. So after saturation, we get a flat line. Please note the sharp turn here. This applies to a single nephron only. If we consider all the nephrons together, the graph is splayed. This is because different nephrons get saturated at different glucose levels. For example, nephrons with less number of transporters get saturated earlier than those with more transporters. Also, nephrons with larger glomerulus filter more glucose. The transport system in such nephrons also gets saturated earlier than those with smaller glomerulus. In short, different nephrons get saturated at different glucose levels. In the graph, initially, no nephron is saturated. So reabsorption keeps on increasing with filtration. At this point, the nephrons start getting saturated. Here, some nephrons are saturated. So these nephrons start excreting glucose in urine. The point from where glucose starts appearing in urine is called the renal threshold for glucose. It's about 200 milligrams per deciliter. In simple words, the glucose starts appearing in urine at a plasma concentration of about 200 milligrams per deciliter. In terms of filtered load, it's about 250 milligrams per minute. Now, although the glucose is being excreted by some nephrons, not all the nephrons are saturated yet. So we can still see some increase in overall reabsorption. But finally, at this point, all the nephrons are saturated. And there is no further increase in the reabsorption. This point is called transport maximum. Normally it's about 375 milligrams per minute. In simple words, the glucose reabsorption can keep on increasing up to this much filtered load. Beyond this, all the additional filtered glucose is excreted. If you are still confused, the graph for excreted glucose might help you. Up to 200 milligrams per deciliter plasma glucose level, no glucose is excreted. Then some nephrons are saturated. So they start excreting glucose. With the saturation of more and more nephrons, the excretion increases. After transport maximum, all the additional filtered glucose is excreted. So the excretion rises parallelly with filtration. But normally we don't need to worry about the loss of glucose, because the physiological range of plasma glucose level is this. And in this range, almost all the glucose is reabsorbed. So these were the concepts related to transport threshold, Splay, and transport maximum. The same concepts are also applicable to several other solutes, like amino acids, phosphate, and paraaminohippuric acid. So this was all about glucose reabsorption. Now let's see the reabsorption of amino acids. At the glomerulus, amino acids are filtered freely. And similar to glucose, they are almost completely reabsorbed in the proximal tubule. There are various transport proteins for different amino acids. In general, entry across the apical membrane occurs by various sodium or hydrogen-driven transporters. And basolateral exit occurs by various exchangers and facilitated diffusion. So this was all about amino acids. Now let's talk about urea. It is freely filtered at the glomerulus. Thereafter, 50% is reabsorbed at the proximal tubule. The thin segment of the loop of Henle secretes 60%. Thick ascending limb, distal tubule, cortical collecting duct, and outer medullary collecting duct are impermeable to urea. Then, inner medullary collecting duct reabsorbs 70%. So finally, 40% of the filtered load is excreted in urine. Now let's see the mechanisms. In the proximal tubule, reabsorption occurs by solvent drag. We have already discussed this mechanism in detail in an earlier video, so we won't go into the details here. Next is the thin segment. You know that the thin segment of the nephron is located in the deeper medulla. Here, interstitial concentration of urea is high. So urea diffuses from the interstitium into the tubule. We will see the reason behind high urea concentration when we study urea recycling under the reabsorption of water. Here we will see the mechanism of secretion only. The secretion occurs by the transcellular route. In the descending limb, basolateral uptake, as well as apical exit, both occurs by urea transporter A2. In ascending limb also, secretion occurs by transcellular route. But the transporters involved are not known yet. In the inner medullary collecting duct, reabsorption occurs by the transcellular route. Apical entry occurs through UTA1, and basolateral exit occurs by UTA3. Talking about the factor affecting urea transport, antidiuretic hormone stimulates UTA1. Thus it can increase urea reabsorption in the inner medullary collecting duct. So this completes urea as well. Now let's have a quick summary. Glucose is filtered freely at the glomerulus. It is almost completely reabsorbed in the proximal tubule. The transport proteins involved are sodium glucose cotransporter at the apical membrane and glucose transporter 1 and 2 on the basolateral membrane. Up to 200 mg per deciliter blood levels, glucose is not excreted in urine. This is called the renal threshold for glucose. At a filtered load of about 375 mg per minute, the reabsorption reaches its maximum capacity. It is called transport maximum. Above this level, there is no further increase in glucose reabsorption, with the increase in filtration. Amino acids are also freely filtered and almost completely reabsorbed. There are different transporters to reabsorb different amino acids. Coming to the urea, it is reabsorbed in the proximal tubule by solvent drag. The thin limb of the loop of Henle secretes the urea by the transcellular route. Then a large portion of the nephron is impermeable. At the inner medullary collecting duct, it is reabsorbed. Finally, 40% of filtered urea is excreted in urine. For watching the video till the end, here is a bonus point for you. In the video on glomerular filtration, we saw that a very small percentage of proteins are filtered. So it seems that we don't need any mechanism to reabsorb proteins, do we? But even with a very small percentage, the total amount of filtered protein becomes very high over time. For example, concentration of proteins in the glomerular filtrate is only 40 mg per liter. But at a GFR of 180 liters per day, the total quantity of filtered proteins becomes 7.2 grams per day. And this is a big number. So we need to reabsorb proteins too. As with amino acids, the proximal tubule plays an important role in this. Talking about the mechanism, the entry at the apical membrane occurs by endocytosis. Inside the cell, they are degraded by enzymes into individual amino acids. The amino acids then exit through the basolateral membrane by various transport proteins. That's all for this video. If you liked this video, please share it with your friends and colleagues too. And don't forget to subscribe because lots more to come. Thanks for watching, see you in the next video.